We're thinking about scale, thinking about scale dependence. Scale dependence is one of those tricky things in working with spatial data or working with GIS or thinking about geography that we just can't beat. Scale, the amount or the scope or the range of something that you're looking at, determines what your answers are. Not just because you might be looking at too much or too little of a phenomenon, not just because your data size or your sample size changes, but looking at things in different scales can arrange and aggregate or separate or build or deconstruct your data sets so that they have different relationships. One of scale's friends is the modifiable areal unit problem, or MAUP, M-A-U-P. Worth a search. Because scale problems tell us that as we're seeking to understand what is happening with the phenomena, with species, cute little bird here, E. gregalis, or with a relationship, that changing scale or changing the size of our analysis area influences our understanding. This is not true just for E. gregalis. This is true across natural and physical and human and societal phenomena. There's box A, range map E. gregalis. In this range map, we can see that 125 of the 258 pixels sampled across E. gregalis's range here have the bird. Red cells, it's there, hanging out, being cute. Blue cells, it's not. This could be a big concern. We sampled a large portion of its range, its expected range, its historic range, and it's missing. This is a bad idea. Wait, I've heard that actually we sampled 116 cells, and the bird was in 79 of them. That's like 2/3. We're fine. E. gregalis is fine. We're not worried about it. We can focus on some other conservation. We can focus on another piece of work. Actually, no, I'm now hearing that 45 of the 45 cells where we sampled, E. gregalis is in 38 of them. Just take it off all the books. We're not worried. It's all good. There's no problem at all. Actually, we're inundated. Of all 17 cells, 17 of them are there. Start making snacks. Go get a menu out. We need to get, this bird's taking over. Which is correct. Well, we probably need to know something about the phenomena we're looking at. What's this little bird's range? What do they eat? Where do they go? What's their territorial behaviors? Right? All these things. We need to understand more about the function and the structure of what this species is doing before we can make a spatial decision about sampling, about presence and absence, about all these different factors. Which cell size is correct? Which grid size is accurate? Well, you probably need to know more to be sure. And isn't that a GIS problem for the ages? Another example here. This is something called the coastline paradox. No shoreline has a defined length because the length of the shoreline depends on the unit that you're using for measurement. So there's the island of Great Britain. If you measure it using fractal units of 100 kilometers, then the coastline length is 2,800 kilometers. See some rough estimates there, cutting some corners and whatnot. But when your unit's only 100 kilometers, you got to make some cuts somewhere. But with a 50-kilometer ruler, the total length is now 3,400 kilometers. It's longer when you use a shorter ruler. It's a paradox. It's a scale paradox. Scale makes us think of the planet in different ways. And when we do that, when we think about the planet differently, when we are confronted with these differences in spatiality, we start to realize that the relationships that we see and observe vary a lot based on how far we're expecting interactions, who we're interacting with, what we're interacting with. This relationship between distance, between location, between how we understand where things or items are, has been observed for a long time. So we can measure and think about these spatial relationships in a few different ways. While scale gives us one lens to look into relationships, and answers vary over scale, another way we can try to understand is thinking about the similarity between observations, the spatial similarities, through something called spatial autocorrelation. This is related to a premise from the great Waldo Tobler, who said in a spatial econometrics paper, trying to understand economic geography, essentially. And in doing the work and the calculations, he basically said, well, if I had a computer fast enough, I'd find that everything is related to everything else, but near things are more related than distance things. Distant things, to enunciate correctly. And if we have relationships, stronger relationships closer, then perhaps we can measure similarities closer or further in a statistical sense. And that's what we can look for. We can look at things that have clustering, positive spatial autocorrelation, where yes, things closer to each other tend to be similar. Land cover, rainfall. Look at census data, you get income level. Negative spatial autocorrelation would have dissimilar values next to each other, a checkerboard pattern. And again, this might invite us to think about some kind of structure, some kind of process underlying this. This is also a process. There's also something happening here when there are clusters of things. Whoops. We use the Moran's eye, which is the tool underlying this, to calculate our autocorrelation, to build up a more rigorous understanding of how things are related spatially. Are they clustering in similar patterns? Are they clustering in dissimilar patterns? And what that allows for us, what that builds in us a capacity to do, is then to make evidence-based and statistically based inferences within our data sets. And that's something we can operationalize in GIS through our interpolation tools.